Ray Bradley is just returning from China this week. He should be hosting again next week, but in his stead, it's my pleasure to introduce David Harning, who's a research scientist with the Institute of Arctic and Alpine Research at the University of Colorado at Boulder. And he's going to talk to us today about his recent Nature Communications Earth and Environment paper and results from multiple Icelandic lake core sediments from the Holocene. Thank you, David.
Great. Can you hear me okay?
Yes.
Great. Thanks to Ray for the invitation to be here and Megan for hosting this morning. As Megan said, my name is David. I'm a research scientist at CU Boulder and also now affiliated with NOAA's NCI paleoclimatology team. And my background is in Arctic sediments and geochemistry. And now, after joining NOAA, I'm also exploring some data science and modeling questions, which is a lot of fun. But today I'm going to be talking about some of the work that started during my postdoc on Iceland paleoecology using molecular lake sediment proxies. And more specifically, I'm going to highlight two examples that I think demonstrate the power of uniting ancient DNA and lipid tool sets. And these two fields are super complementary, but I think right now are rarely used together in paleo studies. What I'm talking about today was part of a much larger project that we had funded by NSF back in 2019, and then ended, at least on paper, last year. And our team's goal was to leverage some emerging analytical techniques, namely lipid biomarkers and ancient DNA from lake sediments, to explore how the Icelandic ecosystem and landscape have evolved in response to late Holocene climate as well as human settlement. And this effort has included a lot of modern calibration work, as well as downcore sedimentary records aimed at reconstructing the greater climate system. And this ranges from temperature to precipitation, and as well as fire and plant communities. But what I really want to just highlight here is that this work has been highly collaborative, and that all these people listed here have contributed to the records I'm talking about today. And I also just want to give a special shout out, though, to Gif and Auswig for leading this project, and then Beth Shapiro and Sam Sacco at UC Santa Cruz for welcoming this collaboration and leading the DNA sequencing effort. So first, I just want to give a little bit of a broader context for why our work matters. As I assume we're all aware, global temperatures are increasing. On the left here, I'm showing ERA5 air temperature reanalysis data, anomalies for the last several decades, where we see that temperatures in the Arctic are warming at a much faster rate than other portions of the globe. And this preferential temperature increase is termed Arctic amplification and is controlled by strong positive feedback loops inherent to the Arctic, such as sea ice and glacier albedo. However, plants also contribute to this. So here on the right, I'm showing recent observed changes in greening of the Arctic from both field and remote sensing data sets. So this greening, or also termed shrubbification, relates to the poleward migration of plants or woody plants as temperatures warm during the summer. And as these woody plants are darker in color, they absorb more incoming solar radiation. And this, in addition to their greater evapotranspiration, also contributes to Arctic amplification. So shrub expansion also has some additional impacts on the climate and the environment. So in addition to impacts on temperature through increased long-wave radiation, shrubbification can also impact local precipitation patterns, CO2 fluxes, additional fuel for wildfires, as well as changes in nutrient cycling that have implications for the broader plant and animal communities that rely on them. However, while we know these processes are occurring today, we don't have firm constraint on the rates of change from the past and what may be expected in the coming century. So recent modeling work shown by here in this paper, Thompson and others, has shown that the impact of including vegetative feedbacks substantially impacts reconstructed temperatures during the Holocene. And by looking into empirical records of shrubbification, say during prior warm times in Earth's history, such as the early Holocene, we can better understand the rates of future change, as well as understand the nuances of shrubbification at different regional scales. And these smaller scales also allow us to address a host of other questions that are related to Arctic amplification, such as the changes in the water cycle and wildfires. So, what do we know about Holocene climate and shrubbification in Iceland? So, our project grew out of a lot of work on Iceland's Holocene paleoclimate from well-dated and high-resolution lake sediment records. So, first, from these records, we know that glaciers largely melted during the early Holocene, and then progressively regrew during the late Holocene in response to decreasing northern hemisphere summer insolation. And then high resolution composite proxy records from West Iceland show that diatom productivity followed a pretty similar trend. And now both glaciers and diatom activity in Iceland are closely related to summer temperature, but both of these tools are qualitative, so they only provide relative temperature changes. Then Holocene pollen records of Betula or birch, its common name, which is the native woodland shrub in Iceland, show that pollen abundance peaked alongside maximum inferred temperatures during the early Holocene. And this is expected as birch distribution today is strongly controlled by summer temperature. However, a major limitation say, pollen is that it's efficiently wind dispersed from thousands of kilometers away. So in this sense, it's really challenging to identify when a shrub is present or absent in the record based on pollen. So while the records here are well dated, as I mentioned, the temperature records are only qualitative, and that pollen is not ideal for constraining shrubbification patterns. So these shortcomings make it challenging to understand the climate sensitivity of birch in Iceland during past warm times. So in other words, the amount of change per degree Celsius. And this piece is vital if we want to place past shrubbification in the context of future climate change scenarios. So one final important piece of Iceland's Holocene history is its relatively late settlement by Norse around 870 Common Era. So this means that in contrast to sites in mainland Europe that have a long history of human influence, the majority of Iceland's ecological history is or has been controlled by natural factors. And in this way, we can isolate how climate has influenced things such as woodland extent. Another point here is that we can empirically test early settlement documentary records. So Landnama book, which is the oldest settlement source for Iceland, was written several centuries after Norse settlers first arrived. However, in this book, it claims that forests covered the shores to mountainsides when they did, implying that there is extensive woodland cover to the higher elevations. And this contrasts with the minimal woodland extent today, which is about 2%, whose reduction has been largely attributed to pressure from humans and their livestock over the last millennium. However, evidence from our research group has shown that the degradation of the landscape likely began before the arrival of humans in response to natural late Holocene cooling. So there's some open questions about how climate and humans, and maybe the combination of them, have impacted the Icelandic landscape and what that means for future landscape recovery. So coming back to our motivation, our project therefore sought to address some open questions on these topics. So first, can we quantify absolute temperature change in Iceland during the Holocene? Then how did shrubs respond to this early Holocene warmth? How did shrubs respond to late Holocene cooling and then human settlement? And then also, what is the impact of lake type and water chemistry on proxy reliability? Now, this latter question was not a primary focus of the project. Proxy reliability became an important second goal as the project got going. And I'll get to later, understanding the impact of redox conditions or oxygen availability was motivated by some new developments in the field, sort of as the project was getting going, and then some unexpected results that we found in our sediment records. And I think with that, many things in research, it's these surprises, which are often the most fun and maybe most valuable to share with you all today as molecular proxies are really growing in popularity. So to answer these questions, we first needed to core some lakes. So on the right here, I'm showing 4 target lake sites here with these yellow stars, where we recovered full Holocene records that we can develop new temperature and ecosystem reconstructions from. And I'll just be focusing on these 3 here from Eastern Iceland today. And then these blue and brown dots show sites where we collected modern surface sediments to develop training sets for a variety of lipid and stable isotope proxies as they cover a nice natural climate gradient. And for each of these four Holocene sites, we also instrumented the lakes with loggers to track seasonal changes in temperature, as well as took seasonal measurements of water chemistry. So just here are a few pictures from our coring campaign, which was in February 2020, which was, I think, a week before the world shut down for COVID. So we really snuck in right at the right time right there. But yeah, for most of these records, we use this Bolivia coring system. And really what I want to highlight here, though, besides the fact that this is a treeless landscape, is that this drive of sediment here on the right covers about 4,000 years of time, and thus illustrates the really high sedimentation rates that are characteristic of most Icelandic lakes. So this lake's total Holocene record would end up being about 9 meters of mud. So about this 3 meters here covers about 4,000 years of time. And in comparison to maybe many of the lake segments records that you all here listening work with, and from other regions of the Arctic, where this 12,000 years of time is recorded, maybe a meter or two, we have upwards of 10 meters, which really highlights the high resolution records that were afforded with to reconstruct climate and ecosystem changes. And our weather was really good on this trip, which for my experience over the years in Iceland is rarely the case. This picture here is from our highland site, our high elevation site in East Iceland, Heiðarvatn, just above the eastern fjords, which you can see here in the back. Again, it was a pretty barren and treeless landscape today at our sites. So let's just take a quick review of some of the tools that I'm going to be relying on today. So first we'll look at our temperature proxy, which are branched glycerol dialkyl glycerol tetraethers, or branched GGTs for short. And these are membrane-spanning lipids produced by a still not super well-known bacteria. But one of their advantages compared to other organic geochemical proxies, such as alkanones, for instance, is that they are ubiquitous in nature, found in every lake. And the modification of their lipid structures seems to broadly relate to temperature across a wide variety of environments. And here's a schematic of their structure where we have N-alkyl chains in the middle connected by ether bonds on the end. And the basis of the branched GGTs as a paleothermometer relies on the number and position of these methyl branches. So the branched GGT shown here has four, and it's termed GGT1A. And this branched GGT is generally found and produced in warmer climates. Under increasingly cooler climates, bacteria then add methylations to these N-alkyl backbones, allowing the cell membrane to adjust its density to different environmental conditions. So these branched GGTs look something like this. This is branched GGT2A with 5 methylations, and then branched GGT3A with 6 methylations. And there's several other structural modifications that can occur with these lipids, including different positions of these methylations, as well as cyclizations that may relate to things such as pH and conductivity. But for today's talk, I think all you need to remember is that we're taking advantage of the number of these methylations, which have been calibrated in numerous studies now to warm season temperature using modern empirical data sets. And at this point, this tool has become quite popular in quaternary sciences, with a number of regional and global calibrations in existence, as well as paleo applications. So on the left here is just one example of those calibrations. This is from Boyoung, Isla Ray, and others from one of their lakes in southern Greenland. And this shows a really nice correlation between MBT prime 5-methyl, which is just one index reflecting the methylations of branched GGTs against summer water temperature. And overall, at the outset of our project, we thought that branched GGTs were going to be a really great tool to reconstruct lake water temperature. However, just as we got going, evidence started surfacing that oxygen availability may also be an important secondary control, and in some cases confound any air temperature signals. So this was really nicely summarized recently by John Rayberg and others, who had a paper come out in GRL that showed if temperature is held constant, the MBT prime 5-methyl index, so the same one down here that responds to temperature, correlates really well with dissolved oxygen. But depending on the region, this can result in either over or underestimates of temperature based on the MBT prime 5-methyl shown here with these different regression slopes. So there's still quite a lot to learn about why this is happening, but changes in microbial community composition are likely at least one factor, and is some of the focus of some work that John and I are working on in the near future. So the second tool that I'm talking about today is ancient sedimentary DNA. So DNA analyses come in several forms, and for our samples, we're using metabarcoding, which is a fairly focused type of analyses where you're looking at DNA from certain types of organisms. So in essence, we take our sediment samples and extract the total DNA in an ultra clean lab facility, which for us was at UC Santa Cruz. And then in step two, we add a primer or tag that preferentially amplifies a certain portion of the DNA. That is common to, say, a group of organisms, which for us were plants and mammals. And this way, we're isolating the DNA that we're interested in and excluding the bulk of DNA, which comes from microbes. And then in step three, we sequence the samples, and then the sequenced data is then compared against reference libraries consisting of DNA sequences that are known from certain plants or animals. And this is where we can identify what DNA is in our samples, and then therefore what taxa were growing around the lake at certain times in the past. And there's other types of DNA analyses around. These include shotgun sequencing, which sequences everything, or bait capture, which is an even more selective technique than metabarcoding. There's still debate as to which method provides the highest reliability and least amount of false positives in your data set. Although I will note there was a recent preprint that I saw the other day from Ingerolsis's group that suggests that metabarcoding may be the best, at least for lake sediments. So just something to keep in mind if you're ever applying this stuff. So one of the biggest advantages of using DNA for our project rather than say pollen is that it has a very local source. So remember that pollen is efficiently wind dispersed, which results in pollen coming from thousands of kilometers away. So just one example is that we find pollen from oak in Iceland's earliest sediments, earliest Holocene sediments, and that tree has never grown on Iceland, at least during the Holocene. So this is really well illustrated in a paper by Sjögren and others in 2016, where they had a lake sediment core analyzed for both pollen and DNA and compared against historical records of conifer or pine introduction over the last century. So times here on the x-axis. So we see that before pine is introduced during the 1960s, shown by this gray bar here, that there's still pollen, albeit in low abundance present, where there's basically no reads for DNA for that taxa. So this highlights the relative reliability of DNA in identifying when certain plants are present or absent on the landscape. However, like every proxy, DNA is not perfect. Some recent studies have shown that its preservation can also be influenced by a number of other environmental factors, including lake water chemistry, as well as where in the lake the sediment is collected from. So an important thing to consider here is that unlike branched GGTs, which really like oxygen, DNA really doesn't like oxygen. So that poses a challenge if we're trying to use both tool sets from the same lake record. So that brings us to the first part of my talk, where we'll focus on a recent paper that we had published in Communications, Earth and Environment. And here we're using adjacent oxic and anoxic lakes to better understand how oxygen impacts these proxies, and then how we can use these two different lakes together to reconstruct Holocene temperature and plant history. So this study focuses on two lakes, Stora and Litla Vidava, from coastal northeast Iceland for a couple of reasons. One is that they deglaciated early in the Holocene. So here on the bottom right is Iceland with the reconstructed extent of the Icelandic ice sheet at about 12,000 years ago, showing that a limited portion of the island was exposed for plant colonization at the time. So this meant we could obtain really long environmental histories from lake sediments, as well as understand how plants colonized and then evolved alongside Holocene climate and human settlement patterns. The second reason is that these two lakes are adjacent to each other, but with very different morphologies and water chemistries. So when translated from Icelandic, Stora means big and Litla means small. And these lakes sit at the same elevation and are only separated by less than a kilometer's distance. But Stora is relatively deep at 48 meters, whereas Litla Vidava is relatively shallow with a max depth of 2.5 meters. They also have very different catchments and lake surface areas. So these differences result in substantially different water chemistries, which I show some examples here on the right. So on top are lake water temperatures over the year, going from October to October. Um, with Stora on the left and Litla Vidavan on the right, we see that unsurprisingly, Litla has warmer peak temperatures during the summer due to its smaller size. And then below this are dissolved oxygen concentrations shown on the x-axis and water depth shown on the y-axis. And just note that the different scales for the y-axis for these two lakes. And what we see here is surprisingly that in Stodovidavat, the water column is well oxygenated and not stratified in either the summer or the winter, which is unexpected for such a large lake. However, it's important to note that our core, shown here, didn't come from the deepest center of the lake here. So there very well may be anoxic bottom waters that occur here seasonally. On the other hand, that we see in our small shallow lake, Liklavidavat, is well oxygenated during the summer and then develops this oxycline during the winter. So this point is important to remember as it plays out in our sedimentary records. So both sediment records from the lakes are relatively long. I'm showing age models here for Stora on the left and Litla on the right. Depth is on the y-axis and time is on the x-axis. We've used both tephra layers, shown here in green, as well as radiocarbon dates, shown in purple, as our age-controlled tools in both these records. And the high density of age control points between these two records means that we have very good age control on the timing of past environmental changes, deduced from the proxies, and the common tephra layers between the records allow us to directly synchronize some of these events in time. And here's just an example of one section of the core from Stodovidavatn. You can see the abundant tephra layers throughout the record. So first, just look at our GDGT data from the two lakes. So what I'm showing here is actually the ratio of two archeal GDGTs over the last 10 and 11,000 years towards present, being 0. And these GDGTs are different from our branch GDGTs in both their structure and their biological source. So rather than bacteria, these lipids are produced by archaea, which is a separate domain of life. And the higher ratio of GDG0 to carnarcheol indicates the presence of archaeal methanogens, or methane producers, which typically occupy low oxygen environments. And this is important as it can allow us to test for the presence of low oxygen conditions and how oxygen impacts the reliability of our branched GDGT temperature records. So while Stora here on the left may have had some potential isolated occurrences of anoxia, we see that Liklabida, on the other hand, has been under low oxygen conditions for almost the entirety of the record from about 8,000 years ago to present. And now here I'm showing the mean summer temperature reconstructions below in black, which are based on our branch GGTs using Icelandic lake temperature calibration that I developed from a lake in northwest Iceland. And these records have all been normalized to the core top, so the temperatures are represented as anomalies relative to today, or 0. And we see on the left that Stora's record fits nicely with the qualitative pattern that I presented at the beginning of the talk, where we have peak early Holocene temperatures, which are followed by gradual cooling towards present. However, on the left, we see a quite different situation, which was surprising, given that lakes right next to each other and experience the same climate. We would expect a similar temperature history. Perhaps even a warmer temperature history from Litla, given its smaller size, as reflected in our modern temperature measurements from the lakes. However, we find a pretty flat record, particularly once the lake goes anoxic 8,000 years ago. Unfortunately, as I mentioned earlier, the mechanisms that cause these branch GDTs to not faithfully record temperature under low oxygen conditions are not really well understood and is an open field of inquiry, but we find this pattern in a number of lakes outside of Iceland as well in high latitude settings. And this is motivating some ongoing work. However, what these two lakes do show is that temperature reconstructions from anoxic portions of the lakes in Iceland, at least for now, are likely unreliable. So now that we have our plant DNA records, here I'm showing some of the dominant taxa we identified, where we have blue taxa representing aquatics, and the green reflecting terrestrial. And these purple diamonds on top reflect where we took sediment samples from for DNA analyses. And then below these are bubble plots where the size of the bubble is proportional to the number of PCR replicates or polymerase chain reactions. And simply we require 2 positive PCR replicates to confirm a plant's present with a greater number of PCR replicates indicating increased confidence in that plant's presence. And importantly, metabarcoding DNA is best interpreted as presence-absence and does not provide quantitative insight into taxa abundance. So in a prior 2023 paper, we used the plant DNA record from Stoda Vidava and focused on the relative delay of colonization of Betula compared to Salicaceae, or Salix, which is another common woody taxa in Iceland, but prostrate rather than erect like Betula. And in short, what we found was a similar delayed colonization pattern across the North Atlantic in a collection of post-glacial lake sediment DNA records. And some first order observations here are that we don't see the same delay in Betula compared to Salix in Lipid. In fact, we see pretty much a lot of these plants occurring right at 8,000 years ago. Another difference is that Lisla has more aquatic taxa, which makes sense given the lake's shallow water depth, which allows light to penetrate the water needed for submerged aquatic plant photosynthesis. And some of these aquatic taxa will come back in a few slides. So these bubble plots are just a subset of the total taxa present, where each lake had about 50 taxa total that we identified. And here at the bottom, I'm plotting species richness, which is just simply the sum of the number of taxa identified in a given sample. And we see at the base of Stoda-Vidavatan's record, we have plants present in nearly every sample and increasing towards present. On the other hand, in Lithos, there are no plants identified for about the first 1,000 years. Yet we know that plants were here because of neighboring Stoda-Vidavatan. But in fact, not only were there no plants found in this early portion of the sediments, we didn't find any amplifiable DNA of any kind. So if we plot over this, the periods of oxic and anoxic conditions inferred from the GGTs, we see that plants appear in Lithlavithavan exactly when the lake goes anoxic. And we know from modern studies that oxygen is really detrimental to DNA preservation, and this shows the same thing in the paleo record. So this is really important because if we chose the Lithla record on its own to infer colonization patterns for Iceland, we would have a totally different history than if we relied on Stola-Vidavatan, which shows the delayed colonization pattern. So in this sense, it's really interesting that Stola, which is never really anoxic, has such a well-preserved DNA record. So this suggests that perhaps the cool conditions at the bottom of the lake, as well as depth of the lake that shields the sediment from harmful UV radiation, may have aided DNA preservation in spite of oxygenated waters. But really just what I want to emphasize here more broadly is that choosing the lake sites really matters for the application of GGT and DNA proxies. And I hope that this motivates some others to consider paleo redox or oxygen conditions more, and that these type of analyses will become standardized in more paleo-environmental reconstructions. And this is important, particularly at the high latitudes where our lakes experience large seasonal changes in stratification and oxygen availability that many of us study. So these considerations are really important through the past as well. Okay, so in addition to the fact that Stola's DNA record showed that birch woodlands may have persisted throughout the entirety of the record, one of the more interesting things that we found comes from Lithlavithavan's aquatic taxa, which are shown here on the left. And notably, we see the disappearance of these two here at the bottom, Isoedes and Calictrici, around 2,000 years ago, both of which inhabit clear and calm waters. So their disappearance suggests that the lake either became loaded with more particulate matter or became more turbid. So looking at magnetic susceptibility, record from the same lake, we see this period coincides with increased mineralogic sediment. Bulk organic geochemistry from this lake, which I'm not showing here, supports this inferred erosion by documenting increased proportions of terrestrial organic matter. And all this occurs before the assumed settlement of Iceland around 870 Common Era. Now, in the Faroe Islands, my colleague Lulay Curtin and others showed a few years ago that the landscape disturbances before Norse settlement were due to prior and less acknowledged occupation of humans, possibly from Irish monks. And this was shown by an increased concentration of fecal stanols produced in the intestinal tracts of mammals, or of ruminants, such as sheep and cattle. So in another paper on Stolavida from our group, led by Nicolo Ardenghi a few years ago, I guess last year, we showed that there was no increase in fecal stanols um, either before or concurrent with known human settlement in Iceland. So this suggested that either humans were present around the lakes, but with limited footprint, or that the fecal stanols were not performing well. And this latter point was considered a possibility, given that next to the most recent portion of the record that shows high concentrations of these stanols, the only other time where these fecal stanols increased substantially in concentration were during the earliest Holocene, right after deglaciation, when there was likely no ruminants present in Iceland. To the best of our knowledge, sheep were only introduced with Norse settlers. So another thing, though, is that these fecal stanols currently lack a proper understanding of how they're produced and preserved in lake sediments. So there's a lot of open questions about how the proxy should be utilized as a tool for human presence reconstructions and paleo studies. To support these records, though, we also had some pyrogenic pHs, which are lipids indicative of biomass burning, as well as some other paleoclimate records from around Iceland. And collectively, what these showed was that it's pretty reasonable to suggest that the landscape instability inferred from our aquatic DNA taxa was driven by natural changes in the climate, including, for instance, some natural wildfires. However, with the uncertainties of fecal stanols, to be sure, we wanted to test with some mammalian DNA. So these are our results. There was nothing. And it was not for a lack of trying, as we tried and tested everything. So we first tried 16S man primer that was successfully used in the Faroe Islands, then two sheep-specific primers, and then a more recent man P007 primer with some human blocking Legos that's been recently used successfully in lake sediments from the European Alps. And all these returned no reads for sheep or any other domestic mammal. And to be extra sure, we then performed some hybridization capture analyses on our shotgun libraries, and this method also failed to yield any reads for mammals. Fortunately, though, we did test all four of these methods on some modern lake sediments from a lake site in southern Iceland, where sheep are really abundant today. And all of these methods yielded positive identification of sheep, as well as detecting bison and a spike positive control for the hybridization capture method. So what this suggests is that our methods are fine, but there was just no mammalian DNA in our records, despite sheep being present in both of these lake catchments today. So this suggests that sheep and human populations were fairly low throughout the entirety of the records, and that therefore any changes in the ecosystem, say reconstructed from our DNA, are the result of natural variations in climate. And so far, this has been quite rare for Iceland, where most downcore records show some degree of human influence. So this makes these records really nice candidates for constraining Earth system models. So to conclude this first part, just a few points. The first being that lipid-based temperature proxies and ancient DNA records can both be biased by low oxygen conditions. As these small lakes are often targeted for paleoclimate studies, we advise careful consideration of your anticipated proxy applications. So maybe your small lakes aren't the best, and there may be some unforeseen complications with DNA preservation. And then we find there's no evidence of human impact on the landscape, meaning that climate has been the primary environmental control on the environment in Northeast Iceland. And while I didn't get to some of the other considerations or conclusions about the paleoecological history from these two lakes, please take a look through our paper if you're interested in that, or feel free to reach out to chat. So the final part that I want to talk about today, real quickly, is about a preprint that we currently have up online on Earth Archive, where this paper focuses on our high elevation site in eastern Iceland to test the range of stratification during the early Holocene and then the near future. So this is what many high elevation regions in Iceland look like today. In other words, mostly cold tolerant heath communities and no woodlands. So by developing records of vegetation from these regions, we can test if woody taxa expanded up to these higher altitudes during times that were warmer than today, and how that may be informative to future expected changes by the end of the 21st century. So to do that, we went to the Lake Hyderabad, which sits in at 422 meters above sea level in the eastern highlands of Iceland. We can see here in this image here that the birch tree line or wood tree line, forest tree line, woodland tree line sits below the lake at about 250 to 350 meters above sea level down in the Breakdaler Valley. And just as we did with Stora and Lillebylva, we monitored the seasonal water chemistry of the lake. So shown here on the left is temperature during the year, with surface shown in red and bottom water shown in blue. Um, Here we see a really nice stratification that develops during the winter before seasonal overturning at the beginning of summer and peak summer temperatures of the lake surface reached in August. On the right is dissolved oxygen concentrations down the water column. So the lake's about 15 meters deep. Concentrations here are shown on the x-axis. And following that seasonal stratification that develops in the winter, we see that the lake goes completely anoxic at the bottom during winter as well. So similar to Stora and Lillebylva, our sediment record for Hyderabad is also quite long, so about 800 centimeters. And as there is no plant macrofossils available to date, we relied solely on tephra layers, shown here individually as green markers. And this record spans about the last 11,000 years, reaching back to local deglaciation as we reached bedrock when we were coring. And the base of this record is also quite minerogenic, shown by these high magnetic susceptibility or MS values, which reflect deglacial sediment from the waning Icelandic ice sheet. So here are our keel GDGT and then branch GDGT temperature records from Hyderabad. On top, we see that our ISO GDGT 0 to Carnarchial ratios that reflect the presence of archaeal methanogens and hence low oxygen conditions increase for the latter half of the record. So this suggests that a substantial portion of the mean summer lake temperature record down here may be unreliable. And it's tempting to ignore this fact as the trends in reconstructed temperature anomalies appear consistent with Northern Hemisphere summer insolation patterns, as well as our other regional qualitative records from Iceland. But to be conservative, we're just going to interpret this first part of the record for now. And unfortunately, this portion comes from the early Holocene when our summer temperatures are highest, and therefore allows us to test the impact of temperature on the local ecosystem at a time that was warmer than today. So then here on the right is the ancient DNA plant record. This is similar just to how I displayed the Stora and Lillebylva records, where on top we have these gray diamonds reflecting where we took individual samples from for DNA analyses. And then below this, we have our bubble plots for some of the most abundant taxa based on reads that we found, where each of the bubbles sizes is proportional to the number of PCR replicates. And then on the bottom here is total species richness shown in gray, as well as species richness for different plant functional groups shown in different colors that correlate to up here as well. So it's here worth noting that despite the oxic conditions inferred from our GGGTs during the early part of the record, the DNA is still well preserved. Now, some of our quality indexes support this notion, meaning that the first appearance of these taxa in the early Holocene should reflect their actual appearance on the landscape. And since the lake is relatively deep at about 15 meters, the preservation of this DNA in oxic conditions suggests that similar to Stora Lillebylva, the depth of the lake and also possibly lack of UV radiation are important preservational considerations. Okay, so now to wrap this up, let's focus on Betchla here. And we're going to compare this with several other stratification records from Iceland to track the early Holocene stratification process. So first, let's just look at some of our quantitative branch GDT temperature records. So here are three from Hyderabad, Stóra and Skóravatn. Skóravatn is in Northwest Iceland, and this is where the downcore temperature calibration comes from. So again, all these records are presented as temperature anomalies relative to today. And it's generally nice to observe that these records are fairly consistent with each other, although the amplitude of peak warming in Stóra Lillebylva here in the light gray It's not quite as high as these other two lakes, and it just may be simply due to the lake's much larger size and more energy required to heat the lake. On the other hand, Skålbotn and Heatherbotn show very similar temperature patterns as well as peak summer temperatures, both at about 3 degrees Celsius warmer than today. And then looking here on the right at some CMIC 6 temperature projections for Iceland for the 21st century. 21st century, we see that the magnitude of warming is within the range of all shared socioeconomic pathway scenarios. So this indicates that our records are good indicators of expected 21st century climate and environmental change. Now, I didn't present this earlier, but we also have measured biogenic silica along with other bulk geochemical proxies from all of our lakes. And this is important for Heatherbotn since the branch GGTs were only reliable for the first half of the record. We can use biogenic silica, which is strongly correlated with spring and summer lake water temperatures in Iceland. We can use this as a relative temperature indicator for changes in the local environment during the middle and late Holocene. And here we see that in accordance with decreasing northern hemisphere summer insolation, relative summer temperatures at Heatherbotn indeed decreased following peak early Holocene warmth. Okay, so now let's look at how birch colonized and expanded across Iceland during the early Holocene, using the available, well-dated, and most reliable records of Betula in Iceland. So in addition to DNA, this also includes macrofossils, which are also sourced locally in the lake catchment, but are just not generally as consistently preserved in sediments as DNA. So first, I'm showing results from the low elevation Torfdalsvatn DNA record, which was published by Inger Alsaas and others back in 2021. And this shows the first appearance of birch shortly after deglaciation around 10,000 years ago. Then at slightly higher elevation, but still coastal, is our Stóra DNA record that shows colonization of birch in northeast Iceland at about 8,500 years ago. Moving up in elevation to Heatherbotn at 422 meters above sea level, we see that birch woodland took another 1,500 years to reach this site. However, interestingly, another high elevation site from northeast Iceland, Nikkavatn, shows that birch had arrived considerably earlier. Although, since the Nikkavatn record does not contain the full record, we're not sure if it occurred earlier than first identified in the lake. In this sense, though, it's interesting to note that there are some really unique microclimate conditions that exist around Heatherbotn in eastern Iceland, particularly related to precipitation. So we're currently trying to develop some hydroclimate records from these lakes to test if precipitation may be another important secondary control on the past distribution of birch in eastern Iceland. But collectively, finally then, the current highest elevation site that is published from 450 meters above sea level comes from the Vestfjordur Valley in Tröllaskagi. And this shows at a similar timing of birch appearance as Heatherbotn about 8,000 years ago. So collectively, these records illustrate how shrubification progressed during the early Holocene. And while the rates of temperature change at this time during the early Holocene are much slower than compared to today, the magnitude is expected by the end of the 21st century, which suggests that the birch woodlands may also expand to these elevations. Compared to the 2% area of Iceland that woodlands cover today, the aerial coverage of Iceland at 400 meters above sea level, which is a conservative estimate or range for our high elevation sites, is about 40%. So that means that a substantial portion of Iceland could be reforested naturally in this time frame. And one final important observation is that birch woodlands were more or less present at Heatherbotn through the Holocene from about 8,000 years ago up until about 750 years ago. And this time, 750 years ago, is coincident with the onset of the Little Ice Age, which is the coolest period in Iceland's late Holocene history. Importantly, this is several centuries after assumed human settlement. So similar to our lakes from northeast Iceland, this timing suggests, again, that climate was the primary factor for driving the ecosystem changes in eastern Iceland, and that contrary to the prevailing paradigm, not humans. So whether this means that humans had a similar or had a limited footprint around these sites compared to other regions, or that the landscape was better managed than previously thought remains an open question. However, what we're sure, though, is that we have three well-dated and high-resolution records from Eastern Iceland that can serve as robust climate ecosystem constraints for Earth system models that are aiming to incorporate vegetative climate feedbacks. Okay, so to wrap up this second part, we showed that vegetable DNA and macrofossil records track the rate of shrubbification in Iceland during the early Holocene. Temperatures at this time reached 1 3/4 to 2 3/4 degrees Celsius warmer than today, and will likely be reached again by the end of the 21st century when compared to CMIP6 model output. And this means that our sites serve as partial analogs for expected climate change. And that if we take the max elevation of our sites at 400 meters above sea level, this suggests that as much as 40% of Iceland could become reforested. And then our late Holocene changes in Betchelo woodlands are consistent with changes in local climate and not humans, meaning that our sites are ideal candidates to constrain Earth system models looking to incorporate vegetative feedbacks. Okay, so just one final point that I want to end with that will be very brief is that we also have microbial DNA from our downpour lake sediment records, as well as our modern surface sediments, shown here in blue and brown. And working with John Rayberg, who's now starting at Claremont McKenna College, and Cliff Bueno de Mesquita, who's in the Fear Lab at CU Boulder, we're using these data sets alongside our microbial GGGT and other lipid data to better probe our understanding of oxygen's impact on branch GGT temperature records, as well as understand ancient microbial communities and how they reflect other society relevant environmental processes. And I highlight this because lipid and DNA data sets are often only skimmed at the very surface, but the amount of data potential is really deep. So for instance, we typically look at about 20 GGGTs in our samples, but there are hundreds to thousands of other interesting microbial lipids that we can scan for and quantify in the lab. And this represents the field of lipidomics. And so I think combining lipidomics with metagenomics, which is the analysis of all other taxa rather than just plants in our DNA samples in paleo records, is one of the really exciting emerging horizons in quaternary science. And I look forward to lots more data coming out of these records in the years to come. So there's really exciting times ahead. So to wrap that up, I hope that these several examples I provide today convince you how much power there is in combining the adjacent fields of lipids and DNA and some of the exciting opportunities ahead. So with that, I just want to thank my collaborators again. This has been such a great project to work on and continue working on. And thank you all for listening today. If anyone wants to chat or is inspired to collaborate on anything presented, whether in our records or your own, my email's here. I'm always happy to chat. Happy to take any questions. Thanks.
Thank you, David. That was a really interesting talk. So usually we start questions with the couple of classrooms that we have. So I think University of Bergen usually goes first.
Yes, I'm really here. Thank you, David, for a very nice talk. Also, a very nice read, the paper that we discussed already earlier today. So we have a few questions for you from the group.
Yeah, hi, Robin here from the University of Bergen. I have one question. It says that basically, given that oxygen is likely, as you said, like one of the many factors affecting branched GD-GT distributions in high latitude lakes, what are the other specific factors that need clarification to fully understand branched GD-GT responses, or other factors that are limiting those?
Yeah, that's a great question. So yeah, I only got to temperature and oxygen availability. But from our modern sample data sets, as well as from culture experiments in the lab, which are not ours, we know that additionally there's pH and conductivity, which are important considerations as well. And I didn't show it in this, but we are able to constrain some of those factors directly or by inference. We're thinking just about past environmental changes. But on that note, it is, I think, also really exciting, in addition to thinking about how we can not just use these tools to understand how, say, oxygen impacts branch GGT temperature patterns, thinking ahead, how do we develop these tools so that we can actually reconstruct these other environmental considerations? Like, so is there a way that with all these lipids, can we simultaneously reconstruct temperature, oxygen, pH, conductivity? And John Rayberg has some calibrations in one of our modern calibration papers, which presents some of those opportunities. So I think, yeah, there's a lot of potential there too. And there's a lot of value in this rather small group of lipids.
Thank you very much.
Yeah, thanks for the question.
You have another question at the moment, or maybe I can ask something very quickly, David. Stein Schepper, also from the University of Bergen. What about the sediment? What's the sediment like in the two lakes? Is it the same? Is it different? Could the sediment play a role in the preservation of the GDGTs and the DNA?
Yeah, so I mean, the sediment's pretty similar. So Icelandic bedrock is predominantly hyoclastite, so volcanic origin. There's not a huge amount of variability, so that mineralogic source is always kind of going to be the same. Given the much smaller size of the Liebigdevattenrecord, there's a substantially higher proportion of organic carbon compared to the larger lake, which is much more diluted. And that is a consideration, or I think it kind of fits in with the oxygen story, right? It's the little lake is loaded with a lot more organic content, which drives or contributes to microbial respiration when the lake is ice covered in winter, which draws out that oxygen and depletes the water column in it, which then impacts the GGTs. So in that sense, I think that factor could impact the GGTs, but the sediment itself, I don't think would impact the branch GGTs or their distributions. Just looking at sort of the broader scale distributions of the collection of branch GGTs that we have, they all tend to look fairly similar, and suggest that most of them are produced in the lake. And what I didn't talk about is there's also GGTs produced in the soils, but those seem to be a rather minor contribution to the sedimentary pool. So I think really to get to your question, I think it's like the external factors, sort of, which is like the lake geometry and DO considerations that ultimately would influence these UGGTs, particularly since the lakes are right next to each other. Now, if you went to a lake in Iceland and compared it to one in Norway, I think the sediment characteristics can be quite different. So that may be a more important factor there, but I haven't looked at that, so I can't really address that specifically right now. But would be interesting to think about.
Yep. Okay, thank you.
Yeah, thanks.
Are there any questions at the University of Buffalo?
Great talk, and you nicely explained how the oxygen concentration influences enzyme sedimentary DNA and GGT proxy, because the two lakes have the different size and hydrology, as well as probably the different microclimate. I'm curious how this difference in hydrology and microclimate might have influenced the GGT and enzyme sedimentary DNA preservation in the lake sediment.
So are you referring to the first two lakes?
Yes.
Yeah, so the hydrology in the sense of like external precipitation should be the same as like climate and temperature.
Probably the catchment size and how much water they receive and the inflow and outflow from the lake.
Yeah, totally. In that sense, we found that it was quite shocking that there was 20 meters of water in our large lake that appeared to be well mixed in both the winter and the summer. And that aspect, I'm still trying to wrap my head around, how there isn't any thermo or oxycline that develops at a shallower depth than what we have. So that lake is, I think, currently a mystery to me, and yeah, a unique example of lakes in the Arctic. But Yeah, I think sort of as I discussed, the stratification, at least in the small lake, that impacts the oxygen availability and particularly the depletion of oxygen during the winter. Yeah, impacts both the DNA and the branched GGT record. Um, so I think that's probably the major consideration. Am I addressing your question right? Sorry if I'm sort of digressing.
Yes, partly. Also, I'm curious, what is the driving factor for the uh, anoxic condition in those lakes during 8 to 1K?
Oh, I see. Okay. Yeah. So at like the beginning of the record, you mean when it was oxic?
When there is anoxic,
When it's anoxic,
yes,
so you're talking about through time, like what would be driving those changes?
yes.
Sorry for misunderstanding. Yeah, so I mean, I think what we proposed in our paper, I mean, we don't know. So, I mean, we just only have to make assumptions based on the data that we have. But what it seemed like there was really low organic carbon concentrations for the first portion of the record, and those increase alongside the change in anoxia in our small lake. So, what we proposed is that for some reason there is just really low organic carbon accumulation for the first 1,000 years in the lake that may be partially due to just lack of substantial plant development around the lake. From the DNA record, we know there wasn't quite a lot around yet. Some basically like salix and some grasses and herbs. So maybe just not a lot of soil development yet. And maybe also just preceding peak warmth, which would have impacted in situ aquatic productivity in the lake. So basically, I think what we likely would have seen is just a shift to larger organic matter accumulation in the lake. And then as the lake is stratified or ice covered it in the winter, due to its salt size, microbial respiration depletes the water column's oxygen, which contributes to anoxia. Does that answer it?
Yeah, thank you. Yeah.
Yeah, cool. Thanks for the
I
question.
have I have another quick question, David, if I may. Thanks for
Yeah.
the really nice talk.
You're welcome.
It's awesome to see these records. Sorry, the owl hasn't found me. No, it did. Hi.
Hi.
I am really curious, just sort of generally as a community. I love that you're looking at all of these different proxies, because I wonder if we can also learn something about the seasonality of production and how that of these different things and how that may change in different lakes or be different between different bacterial or archaeal communities or plant communities. For example, I'm surprised, I don't understand a whole lot about aquatic plants, but I'm surprised that they would be pretty abundant. I'd expect that they require oxygenation in the lake, at least in the summer. And it looks
Yeah.
like there's suboxic conditions in the winter today. Anyway, have you thought about that? Do you have thoughts about where we could go with that?
I have thought about it a lot. I don't know if I have like concrete directions yet. But yeah, the seasonality of the oxygen depletion is very important in stratification. I've talked with Jamie McFarland at the University of Wyoming a bunch about this recently. And yeah, so I think, I mean, right now, our information is mostly, it's basically relying on our modern measurements. So in our lakes, we see that the stratification and oxyclines develop in the winter in Iceland. In Greenland, for instance, that happens in the summertime in a lot of those lakes. So in that sense, maybe if you're reconstructing your paleo redox conditions with GDTs or XRF or whatever, you may assume that in Greenland it's representing a summer signal, whereas in Iceland it's representing a winter signal. Now that's like, I think, a pretty big assumption. Like, what if that seasonality changes in the past, which maybe is what you're getting to? And I don't know if there's a clear way to separate that right now. I mean, maybe looking at like the abundance of GT concentrations at the same time, thinking like if most of these are produced in the summer, we know there's still some production in the winter. So you'd expect maybe lower concentrations alongside. I don't know. Yeah, I'm rambling now. It's a really interesting question. In short, I don't have a great answer, but I think you nailed it. That I think that's one of sort of the next questions is reconstructing the seasonality of that, because that has a huge impact about how this is going to change in the future. There's a lot of moderns, not a lot, but a handful of modern studies thinking about future stratification changes in Arctic lakes and whether that occurs during the winter or the summer, how that influences various geochemical cycles, and in this case, and oxygen conditions, and their flux to the atmosphere and all that. So, yeah, sorry that rambled
Cool.
didn't answer your question, but
Thank you.
happy to chat more if you have ideas.
For sure.
Yeah.
It's 10 o'clock. Let's take one more question from Anna Hughes, who's got her hand up, and then I think we're going to have to wrap it up.
Okay.
Thank you. Yeah, the question is from my colleague.
Hello, David. Will here. Thank you so much for your talk. I hope you can hear me okay.
Yep.
Is that coming through? Great. And I'm a huge fan of your work and really great to see you presenting today. It's a very elegant approach where you're using the multiple proxies to test different theories and ideas. And of course, it reveals a lot about the complications of those proxies at the time. I just had a question, and maybe it's don't worry if it's not answerable in a short space of moment, but I like the approach of using, seeking out relevant climate scenarios in the Holocene, looking ahead to the 21st century and beyond. The one question I had was about the time lags for migration with Betula. Is the assumption that the colonization of the areas that you're looking at is really directly in sync with climate in the early Holocene, or are there other factors that determine location to location barriers, successional pathways, and so on? Because the Betula does seem to hang on then through the longer term Holocene cooling. How confident are you that you're getting a quick climate, that what you're seeing is a climate threshold when you do see it?
That's a great question. And thanks for bringing that up, because I didn't directly address it in this talk, but we did have a 2023 paper in eLife where we looked at the Stoda records. That's the lake that was not impacted by anoxia DNA preservation from Northeast Iceland. Compared that with Torfdalsvatn, which was the lake in central. Here, I'll just bring this back up. So use these two lakes here, Torfdalsvatn and Stoda, which showed that there was some lags with the colonization of these plants relative to their deglaciation times, which were inferred from just the basal ages of the Lake 7 records. And that differed for different woody taxa. So like Salix and Betula were different. Salix came earlier, Betula came later. And talking with some ecologists and biologists, we proposed some possibilities about why these colonization patterns may differ for these different taxa. So climate thresholds are one. Seed sizes is different. So the seeds for Betula are bigger than Salix, so it takes maybe a little bit longer to get across, carried by birds, and so forth. Also, sort of the regenerative characteristics of Salix versus Betula, where Salix is asexual, so it can reproduce just from one plant, whereas Betula requires two. So maybe just a better pioneer colonizer than Betula. So in that sense, I think the earliest colonization patterns are really important after the ice sheet disappears. But then thinking about Hæðarvatn, our higher altitude site, and Nikkelvatn and Vestardalur Valley, Betula was already there by the time those portions of the island melted out from underneath the ice sheet. So in that sense, I don't think it's like the wind or the seed size or necessarily the regenerative capabilities of the taxa that impact its ultimate viability in the new landscape. So in that sense, I assume, and again, places with big caveats, I'm a geologist, paleoclimatologist by training, and not an ecological specialist. So I assume, and somebody can correct me if I'm wrong or there's other information I should consider, but I assume that those higher elevation sites are much more controlled by local climate conditions, whether that's temperature or hydroclimate. So
Oh, great. Thank you. Yeah, sounds like you've got a good handle on it. Thanks so much. Yeah.
yeah, thank you.
Okay, I want to thank David again for the wonderful talk and want to invite everyone to join us again next week on October 29th, next Wednesday, to hear from Helen Innes from the University of St. Andrews in the UK. And thanks very much.
Thanks, everybody, and thanks, Megan.